Tonight we're talking about Chapter 10, the muscular system, or muscular tissue, I guess. It's an overview of the muscular system. We've got one more chapter. Chapter 11 is also on the muscular system. But let's see what we're going to learn tonight. So in this chapter, we will learn about the structure and function of the three types of muscular tissue. We'll examine the events at the neuromuscular junction. We'll describe reactions by which muscle fibers produce ATP. We'll understand the structure and function of motor units and how muscle tension is controlled. And then we'll compare the structure and function of the three types of skeletal muscle fibers. There's three types of muscular tissue, and that's skeletal muscle, cardiac muscle, and smooth muscle. And we've talked about this in the previous chapter. So we see the skeletal, and its function is to move bones. It's multinucleated and striated, and its control is voluntary. In other words, with the skeletal muscle, I think, therefore I am. I can think to move my hand, and my hand moves. I can think not to move my hand, and my hand doesn't move, so therefore it's voluntary. On the other hand, the cardiac muscle, the only place we find cardiac muscle is in the heart. It pumps our blood, and each cell has one nucleus, and it also contains these things called intercalated discs, which we'll talk about later in this chapter. And it's involuntary, so it's what we call autonomic. I can't consciously think, stop beating heart, and my heart stops beating. I can't do it. I might be able to hold my breath, and I can slow down my heart rate, but I can't stop my heart from beating until I get to death. And even then, there may be other factors that involved in stopping the heart from beating, but it's certainly what we call an involuntary muscle. And then finally, the visceral or the smooth muscle, which we find that in our gastrointestinal tract. When we eat, it helps us to move our food through our digestive system. They have one nucleus and no striations. And of course, they're also involuntary. In other words, once I eat something, I have no control over my smooth muscle contracting. What about the functions of muscular tissue? So they produce body movements, they stabilize body position, they store and mobilize substances within the body, and they can generate heat. So functions of the muscular tissue and properties of the muscular tissue are both on page 306. They're just some paragraphs, and they outline the different types of functions and then the different properties. And you can see that, and it continues on to page 307. But there's electrical excitability. One of the things we're going to talk about tonight is something that we call action potential. And anytime we talk about action potential in biology, we will be referring to some sort of electrical energy that's within our body. So yeah, if you've ever heard of the body electric, I think it comes from somewhere else too, but there's actually a really good book called The Body Electric by Robert O. Becker. I've read it a couple times. It's really good. You should check it out if you get a chance. Our muscles, and that's kind of what Becker goes into in his book, is that our muscles are based off of some electrical potential. And this electrical potential, it would be measured in microvolts or millivolts. So it's a very, very small electrical source. So if you think about an electrical source, if you go to your wall outlet, don't do this, but if you were to stick your finger in the wall outlet, you would get what we call electrocuted. But really, that's just the electricity moving through your body, and it comes in at that point, it's coming in at about 110 volts. I don't know, like 60 amps. It probably depends on what kind of a breaker that you have, but the voltage and the amperage are significantly higher in your wall socket than they are in your body. And that's part of the problem with sticking your finger in the electrical outlet, is that it could potentially kill you, because what happens is that electrical potential is going to be greatly increased in your body, and then your heart. We don't talk so much about the heart tonight. We talk about cardiac muscle in passing, and I think it's chapter 20 where we spend more time on the heart. But just know that electrical potential being so low in your body, when you have such an influx of it, like if you stuck your finger in the electrical outlet, it can cause severe damage, and it can really hurt you, and it could even kill you. I mean, I've done it before. I've actually been electrocuted a few times in my life, and I survived it. But I guess it would probably depend on how long being electrocuted for, and how many amps and volts you're getting at that point in time, and also probably a little bit of your physical health too. But anyway, like I said, don't stick your finger in the outlet. Yeah, well, I don't want to go into the reasons I did it, but I have been electrocuted, and I can tell you that it's a very interesting feeling. I've been electrocuted 3 times in my life. And one of the times when I was a kid, I think I did do it on purpose. I saw that thing and I was like, what the heck is this? I stick my finger in there. I never did that again. But then the other times were accidental. Anyway, contractility. So our muscles have to be able to contract. They have to be able to extend, extensibility, and then they also need to be elastic. So essentially they're flexible. In other words, this is a belt. All right. So you can kind of liken it to a muscle, but you see how it's elastic. So when I deform it as such, and when I release the pressure, it snaps back into its original position. So that's the ideal of elasticity, is that the muscle has the capacity to be deformed and then go back to its original shape. Here I'm applying force, and here I'm not. Here I'm applying force, and here I'm not. So when I don't apply the force, it goes back to this circular shape. But when I apply a force, it goes into this ellipse. When I release the force, it goes relatively circular. That's just the idea of elasticity. You probably even find elasticity in some of your clothing. You probably have an elastic band in your underwear. If you have sweatpants, there's an elastic band in that. So, you can stretch those out, and then they snap back into their original position. So let's talk now about skeletal muscle tissue, and we're going to talk about how this is formed. Page 310 at figure 10.2. So what we see here are myoblasts. So myology is the study of muscle. So anytime that you, in your career in biology, that you see the word, or I should say the prefix myo in front of something, that's going to imply that it's related to the muscle. All right, so remember, we've used the suffix blasts in a few different chapters. And a blast is just kind of a form of kind of an undifferentiated cell. So these myoblasts are just basic, like embryonic muscular cells. And then they're going to fuse into this fiber that we call skeletal muscle. And if you notice this big, long muscle here, so you see here they're single celled. But by the time we get to this point, the skeletal muscle is what we call multinucleated. In other words, each cell in the particular muscular fiber, and this is calling it immature muscular fiber, but we can extrapolate this to a mature one. But each muscular fiber that is going to equate to one cell will have multiple nuclei. That's plural for nucleus. Of course, then it points out the satellite cell. So sometimes these myoblasts, they don't necessarily fuse into this muscle fiber. And if that's the case, they might just be resting on this other cell here as what we would term a satellite. So now we're going to go back to page 308, and we're going to look at figure 10.1. So here we see a scanning electron microscope image of a muscle fiber, and then that fiber, as your book terms it, partially unraveled, and then you can see these myofibrils in there. And those are just really just like what we call the striations of the muscle or the muscle fiber. So if we were to actually look at a muscle under a microscope, maybe even in some cases, you may even be able to see it with the naked eye. If you're the type of person that consumes meat, look at it. Next time you go to the butcher, look at the meat and see if you can see these kind of striations. And then if we look over here at the cartoon, we can see the skeletal muscle connected to the bone via a tendon. So you see here that the tendon is connecting the muscular tissue to the bony tissue. And then we've got this epimysium. So think about like the epidermis is the outermost layer of your skin. So the epimysium is going to be the outermost layer of your muscular tissue. And then the perimysium is going to be kind of the middle of that. And then of course, the endomysium, which you see here, that's going to be kind of like the most internal portion of the muscle. So we see there that up here in this above image, well, let's first look at this. So you got the arm here, and you can see this transverse plane going through the arm, and now we're looking at the skeletal muscle via that, and then we're further magnifying that to have even a closer look at it. What we're looking at here is the myofibril, And we can see the myofibril here. And then within the myofibril, we have what we call filaments. And continuing on with the microscopic anatomy of the muscle, going back to page 310, figure 10.2, we can see this a little bit better. So we see what we call the facillae, which is like, it's like, I think of it like a facade. When you go outside the campus, there's a McDonald's, and I think there's a Dunkin' Donuts. I think there's a jerk chicken place over there. There's a car wash. So there's a lot of businesses around the school. You go out and you look at like McDonald's, they've got that distinctive brown roof that kind of arcs out and then kind of comes in like a straight line. That's a facade. So this facillae is just ultimately what's on the outside of the muscle. It's like a sheath that the muscle is within, and it helps to protect that. If we look a little bit closer, we can see once again the myofibrils, and then we'll be talking about these terms in, I think, one or two slides here, the sarcolemma and the sarcoplasma. So those are important. And also, if we look at this lower figure here, another thing that we need to become familiar with is this concept here called the sacromere. The sacromere is basically what we term a muscle cell, where we've got these Z-disc. The Z-discs, they're on the outermost part of the sacromere, and then within that space, there's going to be a membrane, a cellular membrane. But when we're talking about the muscle, it's called the sarcolemma. Instead of the plasma membrane, we call it the sarcolemma. And we'll talk about that here in a few slides. Maybe even the next slide, we'll have to see. Yeah, so here we are. The next slide, as a matter of fact. And this is figure 10.2 continued on to page 311. So this is basically a muscle cell right here. And we can see everything that's in a muscle cell. So it's basically a plasma membrane, a little bit different. So it's not a synonym, but it functions the same as a plasma membrane would in one of your somatic cells, in like your skin, for instance. One thing that you should keep in mind about the muscle is that once the skeletal muscle, the cardiac muscle, the smooth muscle, once all of those muscle types reach maturity in the adult body, they lose the ability to go through mitosis, and that's part of the problem. I remember being down in Florida, and I went to this thing. I don't remember what it was. It was some guy who was like, he worked with alligators, and it was like his whole career. I mean, you could tell he was totally into it. He knew more about alligators than anybody I ever met before. Anyway, when I got a chance to go up and talk to him, he was completely missing his bicep, because at one point in his career, some alligator took a bite out of his arm. And if I get a cut, my skin readily regenerates, not so much with your muscle cells. The adult muscles, they don't want to go through mitosis. And even if they're damaged, they still, there may be a little bit of repair going on. And I think, if I recall, there is a little bit of potential for mitosis in the adult cells, but it's very nil if there is any at all. So if you have any significant damage to your muscle, whether it's an alligator taking a bite out of you, or if you get damaged at work, or if you get damaged in a car accident or something and you lose part of your muscle, probably just going to have to deal with that for the rest of your life, because they don't want to go through. So I'm just illustrating this for you, for one thing, because we're studying the muscles tonight, and then secondly, because I'm trying to outline why we call this sarcolemma versus a plasma membrane, because it is a little bit different, but it basically functions as the same thing. You see this T-tubule here, and these are important because this helps to set up our action potential, which we've got another slide similar to this in more detail coming up here, so we'll save that explanation. Here's our sarcoplasmic reticulum, and you can see these little purple dots inside of here are what we call calcium ions. And like a somatic cell in your skin, you have this cytoplasm, gelatinous material that makes up the inside of the cell that all of your membrane-bound organelles are kind of floating around in. But we call it sarcoplasm in the muscles. So it's basically the same thing as your cytoplasm, but everything is a little bit different in the muscle cell compared to a regular cell. Then, if I was to ask you on a quiz or exam, that this, because it has, this cartoon has 3 nuclei. It could have more or less, but these muscular cells are going to be multinucleated, except for cardiac muscle is not multinucleated and smooth muscle is not, but the skeletal muscle is. So that's a great quiz or exam question right there. True or false, is skeletal muscle multinucleated? True. True or false, is cardiac muscle multinucleated? False. Cardiac muscle is not, skeletal muscle is, so it could be a question. And then remember we were talking about those filaments, and we mentioned the sarcomere, so we see a little more detail, the Z disc right here, and then we've got these thick and thin filaments. The thin filaments are kind of in the, I guess you would call it pink, and then the thicker filaments are a little more, maybe I'd call it salmon color. The Z discs are flanking. They make up this, what we call the centromere. And then this little band right here in the center is called the M line, and M just stands for middle. So you notice here we've got a protein called dystrophin, and then we've got a membrane protein. So the membrane proteins are important because each muscle cell needs to be able to communicate, whether it's with another muscular cell or whether it's with a junction, a synaptic cleft, or a part where it has to communicate with the nervous system, where you get a signal that's telling me to move my arm out or to move it in or to close my hand or open my hand. Dystrophin, so maybe you've heard about the genetic disease muscular dystrophy. So what happens in muscular dystrophy is you have a genetic defect where the dystrophin protein is not forming right. The people who have that kind of disease don't have very good muscle tone, and it makes it, since they have a problem with their skeletal muscle, this dystrophin protein, it makes it hard for them to move. And then we see mitochondria. Of course, there's many mitochondria in the muscular cell, and mitochondria are important because what do mitochondria do? Anybody? I can't see who's here because I'm on my screen, but go ahead. So, is that Sarah?
Yeah.
Hi, Sarah.
Hi.
Go ahead.
The powerhouse of the cell.
Yeah. So since it's the powerhouse of the cell, what can we say that happens there?
It makes energy.
Yeah. Okay. So Sarah's on the right track. It makes energy, or it makes what we call ATP. And ATP is what we call the energy currency of the cell. So very good. So these are important because our muscles, in order for our muscles to move, whether it's skeletal, smooth, or cardiac, we need to have energy for that. So ATP is going to be very important. Therefore, those cells are rich in mitochondria. And then we see this other thing here called myoglobin. And this is just myo, so it has to do with muscle. But this is a way that the muscle can store oxygen, because like hemoglobin is exclusive to red blood cells. Hemoglobin binds oxygen. So these globin proteins, what they do is they bind oxygen. And I think I said it on an earlier slide that the muscle cell can actually storing and mobilizing bodies. So they have the function to store things like blood and oxygen. It's a really fascinating system. It's kind of complex, actually, if you think about it. Okay, so let's move on from this slide. So let's talk about hypertrophy and atrophy. Muscular hypertrophy is the enlargement of existing muscle fibers due to increased production of myofibrils, mitochondria, sarcoplasmic reticulum, and other organelles. So if that's the case, your muscles are going to look a little bit bigger than normal, and then muscular atrophy is a decrease in size of the muscle fiber due to the loss of myofibrils. They occur as a result of aging or disuse. So the idea here is that if you're an athlete and you're a weightlifter, your muscles go into something that we call hypertrophy. When you get to a certain age, or if you stop exercising, that can start to lose some of those myofibrils. And then similarly to that in muscular dystrophy, just to go back to that idea, is that those people who have that disease have a hard time. They certainly can't do what we would call hypertrophy, even though if they're capable, the doctor probably would prescribe a regimen of physical exercise for them, to try to increase their muscular tissue. We're going to talk about the arrangement of the sarcomere now, and this is at figure 10.3, page 312. So I already talked about this a little bit. we got the Z disc. So from Z disc to Z disc, we have what we call a sarcomere. And then we have what we call thin filaments and then thick filaments. So the thick filaments are a little bit darker, pink or orange, whatever you want to call this color. And then the thin filaments are going to be a little bit lighter color. And then in between the M line, which M just stands for middle, so you have a middle line here, you've got this thing called an A band, which stretches here from the H band all the way over to this other H band. We call this whole section the A band, and then the H band is here just kind of in between these. The H band flanks the midline, and then the A band is going to flank the H band, and then the I band, of course, flanks the A band. So those are just what we call zones of the muscle. So when we get to a certain level of study, we can see this more clearly if you have the opportunity to look, or if you've ever had the opportunity to look at the muscles under a microscope, you can actually see these. And then here we just have a cartoon of it. So we can see the thick and thin filaments here, the Z disc, and the entire sarcomere, and then these different bands that we have here in their zone of overlap. So the zone of overlap is important because in the old days, like prior to electron microscopes, people weren't really sure how the muscle contract, but we now know that the contraction and expansion of the muscle, or I should say extension of the muscle, is due to the thick filaments and the thin filaments. You got the zone of overlap, but when you contract, they will go in like that. When you extend, they go out. But there's always a little bit of zone of overlap there. And we'll see this in more detail in an upcoming slide, maybe even the next one. Yeah, very good. So this is table 10.1 on page 313. So we can see this in a little bit more detail. This is a micrograph of the Z discs. We can see our M line here. So basically, if you were looking under a microscope, this is what you would see. So you've got these thick and then the thin filaments, and then you've got the different bands and this H band here in the middle, and these from Z disc to Z disc make up what we call the sarcomere. Page 313, 10.4, figure 10.4. We see the myosin heads and the myosin tail. So myosin is just a protein. Actin is a protein. Well, actually, all these are proteins. Yeah. No, it even says it on the top. I should have read that first. I knew these are proteins anyway. But anyway, let's get back to it. So we have these muscle proteins, and they have certain jobs within your body, contractile. So myosin and actin help you to contract and extend your muscle. The troponin and the tropomyosin, they help you to regulate the physiology of the muscle, and then these other ones, these are the structural ones that help to maintain the structure of the muscle. 10.2 on page 314. You can see here the types of protein and their descriptions. So I'll let you go through this table on your own. There's a lot of information there, but it basically goes through each of those proteins and kind of gives you a synopsis of what it does or its description. And then this brings us to table 10.3 on page 315. So we can see here skeletal muscle. So we're talking about levels of organization within the skeletal muscle now. And we see it defined as just a skeletal muscle. Remember this one right here? We already saw this figure a few slides ago, or I should say several slides ago. So each one of your muscles, your bicep, your pec, your quadricep, each one of those muscles in itself is they would be considered an individual organ. And then each one of those individual organs is made up of muscle fascillae. And we can see that here. All right, so it's kind of this bundle of muscular fibers. And that's exactly what I said, muscular fibers. So that brings us to the next level. And then we can see the sarcoplasmic reticulum, sarcolemma. Remember that from several slides ago? The sarcolemma is like a plasma membrane for a muscle cell. See our myofibrils and our sarcoplasm, and then the nucleus. And in the case of skeletal muscle, I would be more inclined to say nuclei because each one of the skeletal muscle cells is going to have multiple nuclei. And then this just breaks it down even further into the myofibrils, and then the myofibrils are made up of your sarcomeres, which we were talking about 3 or 4 slides ago. Continuing on, so once we get down to even more microscopic detail, we can see there the sarcomere and how the sarcomere is made up of the Z disc and the thick and thin filaments, A band, H band, I band, and all of that stuff. Contraction and relaxation of skeletal muscle, page 316. Myosin pulls on actin, causing the thin filament to slide inward. Consequently, Z discs move toward each other and the sarcomere shortens. Thanks to structural proteins, there is a transmission of force throughout the entire muscle, resulting in whole muscle contraction. Figure 10.5 on page 316. What are we looking at here? 2 sarcomeres? I thought this was actually a pretty cool slide, because what this is doing is it's showing you, so here's your muscle, it's just kind of relaxed, right? And now look at this M line here, and look at how these striations are less intense, maybe even I could say more diffuse. Look at this right here. Look at how much white you see in there compared to the contrast here of the gray. And then if we just kind of move our elbow up at a 90-degree angle, so wait a minute, what's happened here? This H band has became shortened. It looks a little bit more compact, right? I mean, even this gray area with all the white in it, it even looks a little bit more defined now. And then when we look here at flexing our bicep, if we were Arnold Schwarzenegger and we were flexing our muscles, that would be what his skeletal muscle would look like at that point. It would be very compact and dense. And remember, the thick filament and thin filament, they're like this. So on the top, letter A, they're like this, and then letter B, they look like this, and then letter C, they're like this. So they become more concentrated. I believe that what we're looking at here is electron microscope. You should already know this from basic biology, the difference between a light microscope and electron microscope. But the point here is that whether it's light or whether it's electron, there's less light that can pass through when I'm flexed than when I'm relaxed. And even, so let's be careful here too, because even an extension can be like a flexed muscle, right? I can like relax my arm and it's relatively, but I can, and it's a lot tighter just by clenching my fist. So I can still flex my muscle like that, just in a different way, versus if I'm powerlifting and I'm out there modeling or something, you know, like Schwarzenegger. So my point is that obviously when it's contracted like this and those filaments are overlapping, it's harder for the light or the electron to pass through, versus where it's relaxed, it's much easier for the light to pass through. So it's just, you can see the contrast here. This is a really cool slide. All right, so the contraction cycle, and this is on page 317, figure 10.6. Remember, we need ATP, right? So we can see here, we actually have ATP here. So this little blue ball is a phosphate, and I guess your book is just calling this ADP plus a phosphate to show you what happens. Because if we go to step 2, what has happened, hydrolysis happened in the first step. So now this little blue ball, this phosphate, is actually separate from the ADP. And then this myosin right here, this kind of thing, they almost look like uvulas, like a double uvula, and that's able to move at a different angle because this filament. But you see this filament here, this yellow thing, so it's being pulled by this myosin, which is a protein, and that's a result of ATP being hydrolyzed. And then remember lysis to break. What happens when we break a bond? Energy is released. When we break a bond, energy is released. When we create a bond, energy is stored. When we cut that bond, we break that phosphate off of that ATP, and it becomes ADP and a phosphate. We've released energy, and that gives us the ability to flex our muscles. And then once we're done with that contraction, we can see there that the phosphate is going to once again rejoin the ADP, and this myosin is going to go back to its original position, and this filament is going to be released, and that's going to result in a relaxation of the muscle. The neuromuscular junction, or the neuromuscular synapse, parts of a muscle. Oh, so this is, yeah, unfortunately this animation doesn't work, so I don't know what's up with that, but You've got the PowerPoints. You should have been able to download them from D2L. Maybe you could try that on your computer. So let's get back to the idea of the excitation and the contraction of the cells, and this concept connects the events of a muscle action potential with the sliding filament mechanism. So this is 10.7 on page 320 and 321, and these are terms that I've already mentioned to you throughout this lecture. Remember action potential when I was talking about electricity, and I was telling you about when I was a little kid and I stuck my finger in the outlet, and a few other times I've been electrocuted. So this action potential, remember action potential is going to equate to some sort of electrical energy. And in the case of a muscle, sure, it's microvolts, but still it's there and it's important. And this is facilitated by all of these things that we call calcium ions, because remember, an ion is just basically an electrically charged atom that has been either stripped of an electron or has gained an electron. And then that creates the potential for charge, because what it's going to want to do one of two things. It's either going to want to give away an electron, or it's going to want to capture another electron for valence. And you can see here that we've got all of these calcium ions, and now we can see this T-tubulo. We talked about that. Here's our sarcolemma. So remember the sarcolemma, that's basically our plasma membrane on the muscle cell. And then we've got these voltage-gated calcium ion channels here. There's many reasons why calcium is important to your diet. This is another reason, so your muscles function properly, because just think about your muscle for a minute. I mean, it happens instantaneously. I mean, just think about a boxer or somebody who's into the martial arts. When somebody throws a punch at them, I mean, they can react immediately. Or even you, when you're driving your car, have you ever had to slam on your brakes because somebody pulled out in front of you? Well, how fast does that happen? You know, your muscles, you get that signal from your neurons and that synaptic cleft, and it shoots out that calcium ion, and then that's going to go and it's going to react with these sarcomeres, and your muscle expands or contracts. It happens very quick, and you need a lot of energy in there. So calcium is important to your diet. It's good for your bones, and it's good for your action potential in your muscles. So I think that I could make the argument that you could actually react faster if your calcium levels are where they're supposed to be, if they're within that zone, because now you've got these ions. And see here in letter B, the contraction. So here we're relaxed, right? So we see everything's kind of in stasis. And then when we contract, look what happens. Well, these channels start to release the calcium ions, and these are going to react with the sarcomeres, and then that's going to cause a reaction, a contraction. And then this is kind of what we were talking about on that previous slide, right? So this contraction cycle stimulated by the calcium ions right here. And even shows it right here. So very important stuff. Calcium ion binds onto troponin, which in turn undergoes a conformational change that moves tropomyosin away from the myosin binding sites on actin. And then that's how your contraction is going to occur. So there's a little clinical connection here. This has to do with what happens after you die. After about 3 or 4 hours, we get what we call rigor mortis. So mortuary, mort is basically death. Mortgage, think about what a mortgage is, right? Mort means death. So I'm just asking you to think about it. Anyway, after a person dies, or even an animal, like a dog or a cat. Now, it may take less time for a dog or a cat, because obviously they're maybe a little bit smaller, but for a person, about 3 or 4 hours after death, the muscles, they become very rigid, and it's very hard. I've actually tried to move somebody with rigor before, and it's very hard to move. Like if I'm dead and my arm's like this, it will be if I have rigor, it's going to be very hard for you to move my arm. You almost have to like break the arm to move, or I should say break the joint to move it. So what happens is calcium leaks out of the sarcoplasmic reticulum, and the myosin heads bind to the actin, forming the cross bridge. And the cross bridge can't detach since ATP synthesis has ceased. When I teach low-level biology, I define death as the lack of the ability to metabolize. So when you lose the ability to metabolize, then your ATP is not going to be able to be synthesized or broken down for energy or whatever. In about 3 or 4 hours, you're going to go into rigor, and then after about 24 hours, you've got these proteolytic enzymes that are left over, and they're going to digest that cross bridge, and then the rigor essentially goes away. So that's one of the things that if you're into forensics, if you went to a scene where there was a dead body and that dead body was in rigor, then you would know that death occurred approximately 3 or 4 hours prior to that. Yeah, but within that approximately 24-hour window. And again, every body, and I mean this literally, every body is a little bit different. Maybe when your time comes, rigor will set in 4 hours, and then it'll be over in about 22 or maybe 25. Who knows? Just saying that every body is a little bit different. So this is an interesting slide here. On page 322, figure 10.8, the force of a muscle contraction depends on the length of the sarcomeres in the muscle prior to contraction. So what this is saying is that when my bicep is extended, I have more potential energy than I do when my elbow's at a 90 degree. So I can get more force this way than I can this way. And it just kind of shows your optimal length, overstretched versus understretched. But that's the idea, is that it's all about potential energy at this point. I have more potential energy here than I do here for contracting that muscle and getting more force. I can get more force from here than I can from here. I mentioned this earlier in the lecture, the neuromuscular junction, or the neuromuscular synapse. And the events of the NMJ produce a muscle action potential. Voltage-gated calcium channels in a neuron's synaptic end bulb open. This results in a calcium influx. This causes exocytosis of the neurotransmitter into the synaptic cleft. The synaptic cleft is kind of that space in between the synapse of the neuron and then the receptacle or the receiver. In this case, it would be muscle. Neurotransmitter binds to ligand-gated sodium channels on the motor end plate, which causes an influx of sodium into the muscle. This depolarizes the muscle and results in calcium ion release from the sarcoplasmic reticulum. And then the neurotransmitter gets broken down by an enzyme that we call acetylcholinesterase. Yeah, that's a mouthful. Acetylcholinesterase. And that's at 10.9, page 323. So we see there in this little box, here's going to be your neuron. And then you've got these ends here. This is the neuromuscular junction right here. This is going to be your synaptic cleft right here, this light blue. And then you can see there that we've got these pumps that they're going to pump in the sodium ions. And then you've got that ACH that's going to bind to those receptors. They're going to open those up, let the sodium in. That binds the acetylcholine, the ACH receptors in the motor plate. So let's see what happens next. See the scanning electron microscope image of the neuromuscular junction. So the axon, this is going to be part of the nervous system. We see the synaptic bulbs. It looks very cartoonish. It's got that asymmetrical shape. Here's our skeletal muscle fibers. So this is where the sodium is going to be released, or the calcium ions, and then they're going to go transported into one of the T-tubules. So this is the figure I wanted to show you, 10.9, page 323. So this figure just kind of goes through the process of what happens when your nervous system is stimulating your muscle. In other words, like let's say that you're driving down the Eisenhower or the Stevenson, whichever one you drive on, and you're going down 55, 60 miles per hour, and somebody cuts over in front of you, and all of a sudden you have to react. You got to move that right foot over to your brake. Or if you're driving a clutch, or you're driving a stick, you got to like put your left foot on that clutch, and now you're driving a stick. You got two things to react with, right? So it's even more going on at that point. But you can basically see here the ions are released, and these receptors, they're going to open up these gates so the ions can get into the cell. And then this is going to trigger an action potential. So it's triggering some sort of ion release or electrical energy to come in and react with these sarcomeres. And then with an elevated calcium level, this contraction is going to occur, and these calcium ATPase. So ATPase is actually an enzyme that catabolizes ATP. So there's going to be more energy released, and then this tropomyosin slides back into the position where it blocks the binding of the actin site, and then we relax. So that's this slide in a nutshell. I would encourage you to read through these in detail, but that's basically what happens is that your nerve is going to release some ions, and that's going to open up the gates. And then in turn, it's going to stimulate the release of other ions that are going to interact with your sarcomeres, and that's going to lead to a reaction until either you get too tired or either you get some other stimuli telling you to relax. And then this tropomyosin, because remember several slides ago I said tropomyosin was something that helps to regulate the muscles, so that's what's happening there. It's exactly what we were talking about. Remember I showed you that one slide with all the different proteins? Refer to that slide, okay? Muscle metabolism on page 325. Muscles have three ways to produce ATP: creatine phosphate, anaerobic glycolysis, and aerobic respiration. So at least two of those, anaerobic glycolysis and aerobic respiration, you should already be familiar with from the Krebs cycle that you studied in Bio 100 and Bio 111. Creatine phosphate is another concept. Just in case you don't know, typically speaking, whenever a word ends in ACE, it means that it's an enzyme. All right? And I don't know who your low-level teachers were, if they told you that, but I always make sure that I tell my class that, what is it? ASE is an enzyme, OSE is a sugar, and then ATE is a verb, right? My last name, Fugate, is a verb. Kinase is an enzyme. Sucrose, OSE, is a sugar, okay? All right, just a little background there for you. Creatine kinase catalyzes the transfer of a phosphate group from creatine phosphate to ADP to rapidly yield ATP. Because remember, your muscles, they need a lot of energy, and they need a lot of energy fast. And like you're driving down the Eisenhower and that guy pulls over in front of you and you got to slam on your brakes. And if you're driving a stick shift, I mean, man, you got to do that stuff fast. You need that energy. Think of a million other reasons why you might need to use your muscles. Fight or flight, you decide you run, you're threatened, so you run, that adrenaline is going to kick in, and then you've got all this ATP. Well, this thing called creatine is going to help with that. So it's going to catalyze that creatine to ADP, the ATP, so the creatine is going to swipe a phosphate off of the ATP, creates a adenosine diphosphate, and then now we have this thing called creatine phosphate. When the muscle contracts, then that ADP is going to come back and swipe a phosphate, and then that gives the energy for muscle contraction, and then at some point you're going to lose that phosphate again. So this is an anaerobic glycolysis. So like I was saying, this is one you should already know. This should be second nature to you if you're already in bio, what is this, 221? What does anaerobic mean? Anaerobic means no oxygen. Glycolysis, the Krebs cycle, the electron transport chain, yields ATP. So, and that's really, I mean, what your muscle, let's say you're a marathon runner and you're running the long distance, you know. I mean, your body is basically going into glycolysis, and that's going to produce something that we call lactic acid. It even says, too, if you follow this little flow diagram here, it generates a little bit of ATP, but what happens is one of the end products is lactic acid, and that gets into the blood. And lactic acid, like if you go to the gym and you have an intense workout, the next day your muscles are sore, that's because they're releasing that lactic acid. So that's what that sore muscle comes from, is that you know what they say, no pain, no gain, right? Not necessarily true. I mean, you know, if you're in pain, you probably should stop, but when you're lifting weights and then you stop next morning. And I remember one time I was training and I had such an intense workout. The next morning I woke up, I couldn't even sit up out of bed. My abs were just so, And then I had to literally roll out of bed and basically climb up my dresser to get on my feet. My abs were just so, man. But that's the idea here, is that when you're running, when you're doing strenuous work, your muscles, they go into glycolysis and they produce ATP that way, because you can't take in the oxygen fast enough for those cells to go into an aerobic reaction. And that's why when you run or when you do some strenuous physical activity, you become winded, because your body is trying to compensate for all the ATP glycolysis your muscles are going into. So it needs more oxygen to kind of because when we're sedentary, when we're sitting here like this, I'm not winded. I mean, I'm not in so bad a shape that I get winded talking to you, but if I went out and ran around the building about 5 times, I'd probably be winded. And then I'd have to, because I'm trying to take in more oxygen to get it to my various muscles. So I kind of get back to this homeostasis that I'm in right now, where I can talk to you comfortably and I'm not winded. So aerobic respiration, again, you should know this one. And if you notice here right in the center, Krebs cycle electron transport chain. So this is representative of a mitochondria. So the glycolysis has already occurred out here. And then these molecules are going to be somehow transported into the mitochondria. And it's going to go through the Krebs cycle and go through the electron transport chain and generates heat. But it also is going to give us quite a bit of energy here. So see, we get about 30 to 32 ATPs. Right. So that's basic biology right there. You should already know that. I'll read the slide for you just to be thorough. Under aerobic conditions, pyruvic acid can enter the mitochondria and undergo a series of oxygen requiring reactions to generate large amounts of ATP. Basically what I just said. Muscle fatigue. I was just kind of talking about that a few minutes ago. This is on page 327. Muscle fatigue is the inability to maintain force of contraction after prolonged activity. Run around the building 5 times, run a marathon, do a bunch of push-ups, do burpees, any of those kinds of exercises you can do that's going to make you eventually you're not going to be able to do them, because your muscles are going to become fatigued, whether they're out of oxygen or whether your nervous system sending signals to that muscle saying, hey, look, if you keep doing this activity, you're going to get hurt. You got to stop. The onset of fatigue is due to inadequate release of calcium ions from the sarcoplasmic reticulum, the depletion of creatine phosphate, oxygen, and nutrients, the buildup of lactic acid and ADP, the insufficient release of acetylcholine at the neuromuscular junction. Those are things that contribute to muscle fatigue. Central fatigue occurs due to changes in the central nervous system and generally results in the cessation of exercise. You ever been to the point where you're doing such a strenuous workout or an exercise or work at your job, and you're just like, hey, man, I just can't do it anymore. I'm just too tired. You go home, and then you sit down, and you had such a full day at work that when you sit in the chair, you just fall asleep. Well, they think that central fatigue's the idea, what I was kind of just talking about a few minutes ago, about how your nervous system is going to send signals out to your muscle. Or maybe it's the other way. Maybe your muscles are sending signals through your nervous system to your brain and telling you, hey, if I keep doing this activity, I'm going to get hurt or I'm going to die, one of the two, so I need to stop. And that's the idea that they call central fatigue. So the point is it's related to the nervous system, and at some point, you're just going to stop doing exercise, whether you collapse because you pass out. Oxygen consumption after exercise. I was alluding to this a little bit too. Why do you continue to breathe heavily for a period of time after they stop exercising? You have to pay back your oxygen debt. And so really, if you think about it, that's what glycolysis is. You're creating kind of a debt. When early biology classes, we always use kind of the money analogy with ATP, right? You invest like 2 ATP, and ultimately, by the time you go through the electron transport chain, you got about 32 ATP. Well, in glycolysis, if you invest 2, you're lucky if you can get 4. When I run a marathon or I run around my building 5 times as fast as I can and I'm winded, I'm going to be, that's because I'm trying to replenish the creatine phosphate stores. I'm converting lactic acid into pyruvate, and then I'm reloading oxygen onto my myoglobin. Remember we mentioned myoglobin early on in the lecture. Control of muscle tension. Control of muscle tension is on page 328. The strength of a muscle contraction depends on how many motor units are activated. A motor unit consists of a somatic motor neuron and the muscle fibers that it innervates. So what I mean by a somatic motor neuron, somatic is just something that it's a word. When we get to the nervous system, it's basically what we call voluntary. All right, so it's just a voluntary motor reaction. All right, and then activating only a few motor units will generally result in a weak muscle contraction. I can lift up this mouse with much greater ease, fewer motor units, than I can lifting up this book. Activating mini motor units will result in a strong muscle contraction. So that's what I was just trying to illustrate with you, is that my anatomy textbook requires much more force for me to pick up than my mouse does, and therefore I need more motor units to pick that book up than I do to pick up my mouse. So motor unit recruitment is the process in which the number of active motor units increases. The weakest motor units are recruited first, followed by the stronger motor units. Motor units contract alternately to sustain contractions for longer periods of time. And you can see this idea of the motor unit is originating from the spinal cord, because you're getting some sort of signal either from your brain, like, I don't need a signal if I just wanted to pick up my mouse. I don't really need a signal for that. I'm just doing it. But it might be the fact that, I got some external stimuli coming in telling me, hey, pick up that mouse or it's going to fall off the table, or pick up that book or it's going to fall off. You know what I'm saying? How can I put it? If a baseball is coming in, if you're at the ball game, are you a Cubs fan or are you a Sox fan? Let's go to the Cubs game up there at Wrigley. And I'm sitting there. You could be a bleacher bum if you want to, right? We're sitting out there in the bleachers and we're enjoying the game from a distance. And all of a sudden, man, somebody hits a grand slam home run and this ball comes flying at you. And then you're like, oh, you got to move. And so that external stimuli is being taken in by your brain and being sent to your nervous system, telling certain skeletal muscles that, hey, you better move out of the way and you better move fast, or you might get hit by a baseball. So twitches. So if you've ever had a muscle twitch, this is kind of gives you an explanation of it here at figure 10.13 on page 328. The brief contraction of all muscle fibers in a motor unit in response to a single action potential. And then you can see the different periods that your muscle's going to go through for a twitch. So that's basically it, is that when you have a twitch, there's something going on with your action potential that's, I don't know, sometimes I get it in my eye where my eye is like kind of twitching throughout the day, or I've had it here before where this muscle twitches. Or sometimes maybe you just, like if you're sleeping and you just get a twitch. It's just something about your action potential has kind of went awry and caused you to kind of have this moment where you twitch. Frequency stimulation myogram. So this diagram, this figure here, 10.14 on page 329 is what we call a myogram. So notice the difference in the contraction time and force production for each condition below. So here's a single twitch. Here's a wave simulation. Here's unfused tetanus and fused tetanus. You probably have had a tetanus shot before if you get your vaccines. And that just kind of helps it so your muscles don't freeze up, because if you get this fused tetanus, it probably will lead you to death, because your skeletal muscles become paralyzed and you can't move. So you get the tetanus shot because tetanus is found in soil, and it's a result of if you step on a rusty nail or something like that, you can get tetanus, which they say it causes lockjaw, but it can actually freeze up your whole body and lead you to death. So these are just different levels of twitching here on this figure. Muscle tone. Let's talk about muscle tone for a minute. This is on page 330. Even when at rest, skeletal muscle exhibits a small amount of tension called tone. Tone is established by alternating involuntary activation of small groups of motor units in a muscle. So let's talk about these isotonic and isometric contractions. Isotonic tension is constant while muscle length changes. There's two types of that, concentric and eccentric. Let's talk about isometric quickly. Isometric muscle contracts but does not change length. So this is figure 10.15 on page 331. So you can see this concentric movement while picking up the book. And then we've got an eccentric movement while we put the book back down on the table. This is eccentric. This is concentric. And this here is no movement, or what we call isometric. That diagram there is very simple to understand. Types of skeletal muscle fiber. Table 10.4, page 333. So we can see the slow oxidative fiber, the fast glycolytic fiber, and then the fast oxidative glycolytic fiber. So you can go through this table yourself. It gets us to these questions. So what type of fiber does a marathoner primarily use? Well, that's going to be the slow oxidative fiber, because these types of slower muscle contractions are in general considered to be what we would call like our endurance. And then what type of fiber does a shot putter primarily use? Well, that's going to be the fast glycolytic fibers, because it's basically a lot of force in just a very short amount of time. And then the fiber that the soccer player uses is going to be the fast oxidative glycolytic fiber. And all of those different fiber types are located there on page 332. Now you can read all about those. Anabolic steroids, page 334. Synthetic variations of testosterone that increase muscle size and strength may be prescribed for cancer or AIDS patients, among others. Sometimes athletes will abuse those in order to gain an advantage. They have side effects which impact the cardiovascular system, the liver, the muscular skeletal system, tegumentary and reproductive systems, as well as they influence your behavior. I've known some people who were on steroids, and they get roid rage. They've got very extreme moods. Maybe even you think they got anger management issues when really they don't. It's just the drug acting, where they get very aggressive. And it definitely has detrimental effects on your reproductive system. So that's been demonstrated. So it's best to avoid those and just be natural. But in some cases, they may be prescribed for people with certain diseases, and they may help you to have a better quality of life. So let's talk a little bit about the other two muscle types, which we're not going to spend much time on these. For one thing, the chapter doesn't, and secondly, they come up in later chapters in the book, but we need to just kind of outline them a bit so we can kind of keep the three muscle tissue types in context. So cardiac muscle has the same arrangement as skeletal muscle, but it also has these things called intercalated discs. Intercalated discs contain desmosomes and gap junctions that allow muscle action potentials to spread from one muscle fiber to another. Cardiac muscle cells have more mitochondria, and their contractions last 10 to 15 times longer than skeletal muscle contractions. So if you can remember desmosomes and gap junctions from chapter 1 or chapter 4, I can't remember, but it's just ways that cells are held together. So they've got a little bit more of a loose fit, but they need a little bit more space so that action potential can jump. And that's what kind of creates the heartbeat, right? Because it's all based off of, well, it says it right there in the book, action potential. It's all based off of electricity. Action potentials jumping from cell to cell are going to give that heart its contraction, and the heart has to constantly beat, because if it doesn't beat or contract, and you're just not going to be able to survive. So it's very important. So it's a little bit different game than skeletal muscle, but cardiac muscle is similar to skeletal muscle. I like to think of it like this. Cardiac muscle is a little bit more refined than the skeletal muscle. It's almost like, it's like my Toyota. I got like kind of just a cheap car that's got really good gas mileage. But I got a friend, he's got these muscle cars. I mean, it's just, he spends time working on his car, and he takes pride in it. And man, that engine's just like, it's finely tuned. Where my Toyota is just yeah. As long as it's running, I'm happy. I know it's a bad example, but skeletal muscle versus cardiac muscle. Cardiac muscle there's more at stake. If an alligator bites my arm, bites half of my bicep off, I can still live, right? I can still go on with life. But if that alligator bites my chest and eats my heart, I'm going to have a hard time surviving then. So there's a little more at stake with your cardiac muscle. Smooth muscle looks quite different than cardiac muscle and skeletal muscle. It is thick in the middle and tapered on the ends, and it is not striated. It can be arranged as either a single unit or a multi-unit of fibers. And there's a nice diagram of that there on 1016 on page 336. And this is another same figure on page 336, 1016. Smooth muscle contractions start more slowly and last longer than skeletal and cardiac muscle contractions. Smooth muscle can shorten and stretch to greater extent than skeletal and cardiac muscle. Smooth muscle fibers shorten in response to stretch. So I guess the gist of it is that the smooth muscle is a little bit more relaxed. It's also involuntary, and it's a little bit more elastic than your other two types of muscle. So here's a summary of the types of muscle on page 337, table 10.5. A few more muscle facts. So the regeneration of muscle cannot undergo mitosis, hypertrophy, hyperplasia, and smooth muscle pericytes. Well, we already talked about all of this stuff earlier on in the chapter, so you can find out more about this on page 337, 10.10. And then it goes up a little bit into the embryology of the muscle there on 10.17 on page 339. But most muscles are derived from the mesoderm, which develops into the somites. So you got the myotome, the dermatome, and the sclerotome. And so we can kind of see how it forms here. And we've got this transverse cross-section we're looking at here. So here's the somite, and then this is this developing nervous tissue around it. Here's going to be your muscle in here. And this figure is on page 339, which I already said that. Aging in muscular tissue, page 339 again. About 30 to 50, lose about 10% of your muscle tissue, and it's replaced by fibrous connective tissue. And then also includes some adipose tissue, but by the time you're 50 to 80, another 40% of the muscle tissue is replaced. The consequences are muscle strength and flexibility decreases, reflex is slow, and the slow oxidative fiber numbers increase. Okay, class, well, that's it for the PowerPoint tonight.